Hi everyone, welcome back to another anatomy episode. In today's video, we are going to go over cytology. In other words, kind of just the study of cells and the study of cell anatomy and all of the organelles that are encompassed within it. So please like and subscribe for more content in the future. And now, without further ado, I'm going to get into the video. So the first thing that we are going to look at is the phospholipid bilayer. In this bilayer, in kind of this like turquoise color over here, we have the integral protein. Integral proteins are basically going to form pores or channels that allow smaller water-soluble molecules as well as ions to pass in and out of the cell. So anything that isn't able to diffuse through the phospholipid bilayer, it is going to go through an integral protein. Then over here in purple, we have peripheral proteins. And peripheral proteins are usually found along the cytosolic surface of the membrane. So basically, kind of like the inside surface. And it is usually right next to an integral protein. Then we have cholesterol in this light blue color over here. It is this structure where you see like a couple hexagons and some squiggly lines over here. Cholesterol is basically going to help with the fluidity of the plasma membrane so that it is able to kind of like move around and not be super rigid. Then each one of these little like balls with a tail, that is a phospholipid molecule. We will get more into the properties of phospholipid molecules in the next slides. Up at the top over here in yellow, we have a glycolipid. And then in pink, we have a glycoprotein. The way that you can differentiate between a glycolipid and a glycoprotein is because the glycoprotein is going to be connected to this little bump over here, which is a protein. So you can see a bump here, you can see one of the bumps here. If it's connected to that, then it's going to be a glycoprotein. If it is not, it's a glycolipid. And then finally, again, this is a phospholipid bilayer, is this whole thing where you have the phospholipid molecules creating kind of like this sandwich effect. Moving on, we have the chemistry of the phospholipid bilayer. So over here, we have the phospholipid head in pink, and the phospholipid head is basically going to be hydrophilic, meaning it is going to be facing the external environment where it can be in contact with water or other solvents. Then we have the phospholipid tail here in yellow. The phospholipid tail is hydrophobic, meaning it has a fear of water or of solvents. So basically the way this works is the phospholipid head is on this exterior surface over here and also over here. And then because the tails, the phospholipid tails are hydrophobic, they are going to be conditioned and they're going to be sandwiched in between so that they are hiding from the water or from the solvent. If you look at this last bullet point over here, it explains it. Tails are hidden within the bilayer in order to avoid contact with the solvents or water. So they have a phobia of it. They don't want any contact. And that is basically how the bilayer is created. Now we're getting into more of the cytology portion. And again, cytology is the study of cells or cell anatomy. So over here, we have a lovely diagram of just a basic cell and everything that would be included within it. So over here, we have your smooth endoplasmic reticulum or the smooth ER. In the center, this pink portion over here, this is your nucleus. And within the nucleus, over here in this light blue, it houses the nucleolus. On the outside exterior portion in green, you have your rough endoplasmic reticulum. And you know that this one is the rough ER because it has all of these tiny little dots over here. These tiny little dots are known as ribosomes and they help with protein synthesis. So I labeled the ribosomes, I labeled two of them over here in red. These are your ribosomes. Next, you have your mitochondria. This is where majority of the ATP in the cell is produced. ATP, also known as adenotriphosphate, basically is just like the food and the energy for the cell. So you see one mitochondria here, you see another one here, and a really big one over here at the front. Then these churro-looking things in this light aquamarine blue, this is a central, and around it you have these little like microtubules. This in orange is a lysosome, and in blue you have peroxisomes. So lysosomes are always going to be bigger than the peroxisomes. Next, this structure over here in pink, and it's actually this whole structure, but it's just not all highlighted. But this whole structure over here is your Golgi apparatus, and we will get into the function of the Golgi apparatus in the next slides. But you can see that kind of like on the edges, they kind of like come to a pinch and form these circles. These circles are going to become vesicles where different compacts of the cell are going to be transported to the plasma membrane and then released out of the cell. So that is what these kind of like circles at the very end are. And finally, you have your cytoskeleton over here in darker blue. This is basically just helping with the structure of the cell, keeping that nice circular or oval shape of it. We have your microvilli, which I drew in over here because it wasn't included in the diagram. But the microvilli kind of just help propel substances along the surface. And finally, the encasing of the whole cell is your plasma membrane. So now we're going to get a little bit into the function of all of these cellular organelles. All of the different factors that I have gone over, such as like the nucleus, the ribosomes, mitochondria, those are all considered to be organelles. So your nucleus is the control center of the cell. It helps with all of the genetic material, the DNA, and the nucleolus. It is basically the cookbook for how everything in the cell is supposed to function and work. Then you have the mitochondria, also it's commonly called the powerhouse of the cell. This is where majority of the ATP is produced, and again, ATP is the energy that the cell uses. Next, your Golgi apparatus. It's basically a series of tubules with ribosomes that are attached to it, and it is responsible for the synthesis and packaging of glycolipids. So remember how I talked about how it has those vesicles at the end of it? Again, these structures over here, the vesicles, those are going to pinch off, go to the plasma membrane, and they are transporting those glycolipids that is said in the definition. Then you have your rough endoplasmic reticulum, or your rough ER for the abbreviated version. This is basically responsible for the synthesis and packaging of proteins, while the smooth ER is responsible for lipids and steroid hormones. So rough deals with proteins, smooth deals with lipids and steroids. Then you have your ribosomes, which is protein production, which is why the ribosomes are attached to the rough endoplasmic reticulum so that it can help with that protein production and packaging and synthesizing it. Next, the last couple set of organelles that we have your cilia. The cilia is basically going to have this propulsion mechanism, also known as just like rhythmic beating, and it propels substances along the cell surface. So you actually have cilia within your nose, and it helps to get rid of any dust or debris or pathogen that might be trying to enter your body through your nasal canal or through your nose. So it helps with immunity support. Then your lysosomes. Lysosomes have very strong acidic digestive enzymes within them, and it basically helps to break down damaged organelles or phagocytosed pathogens. So basically, again, helping with your immune function. Then your microvilli. Microvilli are those extensions. They basically increase surface area for absorption. So the higher your surface area, the better you can absorb. So you want to have lots of ridges and lots of folds within your cell or your cells or other bodily structures. Finally, your central. Your central helps with chromosome movement during cell division. So, if you guys have heard of mitosis or meiosis in molecular biology, then the central is a key aspect of those processes. Finally, once again, your cytoskeleton, it helps with strengthening and establishing the cell interior and the shape and the structure of the cell. And finally, peroxisomes. They contain oxidative enzymes that help break down those fatty acids and ethanol. So lysosomes help with like immune function against pathogens and damaged organelles, and peroxisomes help to break down fatty acids and ethanol. Ethanol is also known as alcohol. So that was everything for this lesson on cytology. I hope you guys found it helpful. Please, again, like and subscribe for more content. Leave your comments down below with questions, concerns. If you want anatomy tutoring, I offer that. As well as if you have video recommendations, I am also open for that as well. Thank you so much for watching, and I will see you next time. Goodbye and have a great day.